Okay, hello everyone. Welcome to this third lecture of our special lecture series. So my name is Min Zhang. I'm a professor of statistics and the associate director of data science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded R25 summer workshop on big data training for cancer research. So today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped setting up all the Zoom meetings and handling all the registrations. So he also records the lecture and posts the recording at our workshop website later. So now I would like to introduce our webinar moderator, Dr. Nadia Lama. So Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. She has been the manager of the Purdue site of the Collaborative Core for Cancer Bioinformatics since 2015. Dr. Lama has a background in biochemistry, molecular biology, computational biology, and she got her PhD in 2015 from Purdue. So her research interests include single-cell data analysis and data integration with a focusing on immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Thanks, Min. So welcome back, everyone. And just as in the other special lectures, we will remain muted throughout the duration of the lecture, and then we will take questions after. So feel free to post any questions that you have in using the Q&A function at the bottom of your screen, and then we'll go through them at the end of the lecture. Feel free to upvote questions that you're particularly interested in seeing or in having answered, because those will bubble to the top and they'll have a greater likelihood of being answered. So thank you all, and I'll hand it back to Min now.
Thanks, Nadia. It's my great pleasure to introduce our speaker today, Dr. Stephen Chanock. So Dr. Chanock is a leading expert in the discovery and characterization of cancer susceptibility regions in the human genome. So he has received numerous awards for his scientific contribution to our understanding of common inherited genetic variants associated with cancer risk and outcomes, including Niehaus, Salzer's Wisbeck Award in Clinical Cancer Genetics, and the 2015 Jeffrey M. Trent Lecture, the 2021 NIH Clinical Center Contemporary Clinical Medicine Great Teacher Seminar. He is an elected member of the Society for Pediatric Research, the Association of American Physicians, and the American Epidemiological Society. Dr. Chanock received his MD from Harvard Medical School in 1983 and complete clinical training in pediatrics, pediatric infectious disease, and pediatric hematology, oncology, and research training in molecular genetics at Boston Children's Hospital and the Dana-Farber Cancer Institute in Boston. So since 1995, Dr. Chanock has served as medical director for Camp Fantastic Exit Disclaimer, a week-long recreational camp for pediatric cancer patients, which is a joint venture of the NCI and Special Love Incorporation. So today, Dr. Chanock will speak on a story about Chernobyl accident in 1986. So please join me in welcoming Dr. Chanock.
Thank you. Thank you. And thank you very much for the very kind introduction and the invitation to speak. I'm going to talk about sort of an integrated analysis of looking at where and how we can use our tools to understand very important public health crises, such as the Chernobyl nuclear power plant accident on April 26, 1986, and how and in what way risk of cancer actually takes place. And I'll go through a little bit of the epidemiology, and then we'll talk about the most common cancer seen after that, thyroid cancer. And then a different study I'm going to talk a little bit about, story number 2, we're using genomics to understand a very important public health issue of the transgenerational effect. And each of these stories they were just published in Science last month. And there are several more papers to come out over the coming year or so. So you'll get a combination of published data and some new things that we're looking at and thinking about. But I think for bioinformatic opportunity, it's really quite something to be able to look at the germline, to look at the somatic and the environmental all together, okay? So we know that there were literally millions of people with different degrees of exposure to the radiation after April 26th, 1986. And the star shows you where in the northern part of Ukraine, the actual explosion took place. And the key issues to recognize are that these maps show where there were high doses, particularly the redder. And then there were 2 different types of exposure. One was residential exposure. Those are the people that live there, and their environment became contaminated. So the radiation, the radioactive nucleotides fell into the fields and were part of the grass. So the cows were eating those, and then the children were drinking the milk from those cows. So there's a very clear chain of unfortunate transmission. But then there are the occupational exposure, the so-called liquidators, and that will be in the second study that I'll talk about, those who were brought in to clean up. So this health effects question has been one that's been very much on people's minds since the late 1980s. And the National Cancer Institute, where I am, has had a strong commitment to this. And I just wanted to sort of point out that there are some giants and people who have been thinking about these things for years. And Gil Beebe, who there's a seminar every year at the National Academy of Sciences on radiation epidemiology, and Elaine Ron. Each of them are black and white because unfortunately they're no longer with us, but they were really marvelous pioneers. And our colleagues in the Ukraine. What I'm going to present to you are 2 studies that have taken about 8.5 years each to come to fruition. A lot of politics, a lot of sensitive issues, and then of course the expertise here in the NCI who've really been instrumental in these kinds of studies. So the first one is story number 1, is the environmental radiation exposure and genomic alterations in papillary thyroid carcinoma. So we know that papillary thyroid carcinoma has a high incidence in women, about 95% survival in the West, a little bit lower in the Ukraine. It's a cancer of young adults, somewhere between 20 and 45, highly differentiated. A mutational burden is higher the older the patients. I'll talk about that again in a minute, about the Cancer Genome Atlas has done an analysis of them. And there are 2 particular mutations in BRAF 600, a point mutation in the TERT gene in the promoter. These can drive therapeutic decisions here in the United States and in Europe. So it's one of those unusual circumstances where what causes it can also be very important for the prognosis per se. So back in 2014, the Cancer Genome Atlas, so-called TCGA in Cell, published a large landscape analysis of 496 papillary thyroid cancers. Well, a few of them turned out not to be that, as we could see both from the genomics, which often doesn't lie, as well as a re-review of some of the samples, but they were all sporadic. And they were all, by and large, much older, 45, 50, 60-year-olds. And this is important. So when we look at the so-called commute plot, this is sort of a bioinformatic approach of jam it all together. So do you see sort of cool patterns? And to me, these are visually very compelling, and they give you hypotheses that then you go after. And so here, I call your attention to the significant mutations. Here in this TCGA study, about 82% could they identify at least one driver. And most of the cancers had just one driver, unlike smoking lung cancer or UV-induced melanoma, where you have multiple hits that are going on over 30 years. And so you have these clones that keep acquiring new mutational events due to the insult not being stopped. So for sporadic cancers, you often don't see quite as much subclonality. So here the key issue is, if you look, the vast majority, 60% had the BRAF and another 10% HRAS. So point mutations were accounting for a large fraction. And then there's a smaller fraction of fusion oncoproteins that were identified by RNA-seq, confirmed in the DNA, and that represented 15% or so. And then relatively quiet with respect to SCNAs, and overall the mutation rate is relatively low. So what do we know about exposures and somatic mutations? Well, not a whole lot. We know about cosmic signatures, but when we actually go look at cancers, how well do we do that? Well, we don't do particularly well, simply because TCGA and ICGC really paid very little attention to the exposures and the environmental drivers. 8 years ago or 9 years ago, I was the acting director of TCGA for a year, and it was already too late to try and solve that issue. So, but Chernobyl this is when this whole idea was born in my mind. So we're paying a great price in trying to work backwards, and it's incomplete and expensive endeavor. In the same way, going forth clinically, it's a highly biased set. So when we do our analysis, we have to be really careful. It's the way in which the tumors were ascertained. There's a very profound bias. Size really mattered. So you had late stage, larger tumors. So you're not looking at the spectrum of tumors, but of particular types. But we have things like mutational signatures, the cosmic signatures, whether you're using MutSig or SigMut, or there are a number of different approaches. These can be very useful forensic tools for epidemiology. And most of our tumor sequencing is sort of like, NCIS or Law and Order on television. You come in, you find the murder victim, you do fingerprints, and you try and work backwards. That's what most of tumor sequencing is. And then you say, what can you infer about endogenous versus exogenous processes, and what kinds of select exposures are there? And only a small fraction can we really make that happen. So one of my points going forward, all the trainees and people who are doing this, is we should never forget the context of the cancer genome analyses. Cancer genomes don't occur in a vacuum. They're not homunculus cancers. They are individuals who have lived, breathed, eaten, done smart and dumb things, smoked for 30 years, drank the wrong water, you name it. And that is really important in our interpretation. So why this is so important is, let's go back to Chernobyl. So there have been multiple studies that have looked at this really key result, both in Chernobyl and in children who had been irradiated for another disease like cancer, or in some circumstances before, seborrheic dermatitis and scoliosis. This pooled analysis clearly showed that age was really profoundly related to that. The younger an individual was when they were exposed to radiation, the greater the likelihood is that it would have an effect, and actually the effect would be measured as thyroid cancer. So we know that there's this age question on the left, as you can see the difference between the two curves, and on the right, the pooled analysis show that that age and dose are very important to a point, and then they start to tail off. Okay, so if we go back to the way we think about what I'm going to now talk to you about is Chernobyl accident, reactor 4, terrible crisis, the weather comes, spreads it all over, and then lo and behold, you have the milk supply contaminated in young children. Remember, this is the Ukraine 1986, still in the Soviet system. Food opportunities were quite restricted. Diets were, the number of different things that people would eat could be counted on both hands, and that was it. You didn't have Uber Eats and, let's go out and get whatever we want. Very, very set, restricted diet. And unfortunately, milk for young children was central, and it still is, but with respect to the I-131, which is the radionucleotide that we could easily measure and track. There are other radionucleotides, probably in smaller amounts, but the I-131 was the one that was most widely distributed when you look at the dosimetric charts after Chernobyl. Okay. So when we look at the age of PTC, which is papillary thyroid cancer, and radiation dose distribution, we are looking at cases that are occurring, almost all of them under the age of 40, as you can see. And the exposures were anywhere from 15 to 25 years to 30 years before. So this is really pediatric exposure and then developing this disease as a young individual. If I were to put on the map here what TCGA would look like, TCGA would start here and go way out here into the 60s, okay? So the mean was 29.7, and you could see that there was a wide range of estimated doses. And there's a whole science of dosimetry, of how you estimate and what the uncertainty and the statistics of that using Monte Carlo simulations and different kinds of models. So we know, because of the cohorts and the individuals who we chose to actually analyze, with pretty high accuracy, how much radiation they're exposed to. And we purposely went and identified 81 individuals who were born after the accident and a little distance from that area who did not have high exposure. So we had at least something comparable in the Ukraine, because age is very important in this. And there were some individuals who had 1,000 milligray, 1 gray, 2 gray, up to 9 gray, which is really a fatal amount. But surprisingly, the person did not develop what you would expect in a nuclear accident or nuclear exposure, to high dose radiation, something like this: leukemia, bone marrow failure, gastrointestinal failure, skin sloughing, and a lot of fatality associated with very high doses. So one of the remarkable things about this is that it's protracted medium to low doses that we're able to study. Okay, so here you can see the dose distributions. So the flow of our data, we went to the Chernobyl Tissue Bank. This is an international UN overseen group that's based in the UK at Imperial College under Jerry Thomas, a wonderful colleague and collaborator. They have a panel of experts who review the pathology from Japan, experts from Hiroshima and Nagasaki, where the Radiation Epidemiology Research Foundation follows the individuals who survived the atomic blast back in 1945, where they were very strong investment in understanding radiation effects there. And we identified 440 confirmed cases with fresh frozen material. 431 had matched normal, some of which were non-tumor thyroid and or blood. We then did the tissue sample with a pathology review, the RNA and DNA isolation according to the standard TCGA-like protocol, the sequencing, the expression, CNA, LOH, epigenetics, genomic analysis. And what's different here is, for the first time, we put dosimetry, radiation exposure, on top of this for an integrated analysis. Honestly, TCGA always stops at this genome analysis, and most of the papers that you still read to this day in Cell in Nature are talking about genomic analysis, but not integrative analysis. So for this, we had the SNP chip from Illumina, the methylation chip from Illumina, microRNA sequencing, mRNA sequencing, and whole genome sequencing, 80x for the somatic and 30x for the normal tissue. And this is what our paper published last month, led by Lindsay Morton, whose picture you saw was really essential, is the effect of radiation on genomic features. And that's what I'm going to talk about. So here's our landscape plot, and we organized a little bit differently. So you can see we have unexposed individuals, those who had 1 to 99, which is very low dose, those who had 100 to 199, 200 to 500, and above 500. One of the things that's striking is, if you look at the driver mutations here, both the unexposed and the 1 to 99 look very similar to what we'd seen in TCGA. But as one starts to move towards higher doses, you see the shift towards a greater fraction of being fusions. And that's one of our conclusions. And how and in what way those fusions develop, we can actually look at sequencing and not only infer, but we can understand one of the exciting things is how radiation actually causes the cancer here. And we're going to talk about that. Okay, so the distribution of mutational burden in cancers, we know that the adult thyroid is a quiet one, sort of like other pediatric cancers, where you are, somewhere in the range of 1 mutation or less per megabase, per 1 million bases. Smoking and UV driven are 2 to 3 orders of magnitude above that, 100 to 1,000 times more. Continual bombardment of that. Interestingly, R is on the end here, the Chernobyl, where we had a discrete amount of radiation and then it tailed off or individuals moved away. So here, in terms of mapping, we can look at this not as a continuous problem that you would have with smoking and UV, because almost nobody stops smoking or stops going outside with melanoma and previously with lung cancer. Here, we knew how long people were exposed as well. So we can see there's a very low background mutational rate, which tells us that there's probably very little activity from the radiation at the single base level. So when we started to look at the signature analysis, going to Cosmic for the catalog of somatic mutations, as you know, there's 6 possible base substitutions, looking at the 96 possible combinations. There are multiple programs to do this. And when we did that, we know that you can look at certain kinds of signatures, like the tobacco signature. Signature 4 is very classic with that. Signatures 1 and 5 are the clock background. So when we looked at our single base substitutions, we could see that we saw almost exclusively the clock, SBS 5 and SBS 1. There's an unknown SBS 8 of about 15%. And then we saw a little bit of the apobec, which is important in terms of response to sort of primitive organisms, viral and parasitic infections, and minimal of the reactive oxygen species. If we look at the 8, I'm not going to show you here, but the 8 has nothing to do with radiation dose. So the way in which we're able to do our analysis, because we have that environmental exposure, is to do logistic regression analyses and ask the question, does something change with increasing or decreasing radiation dose? And the relationship between the radiation dose and the presence of SBS 8 was completely non-significant, not interesting at all. So we conclude that we don't see an SBS signature that's correlated with radiation, which is not surprising from what we know in the previous literature, okay? So here, when we started to look at the Cosmic, which has gone to a second level, so to speak, of insertions, deletions, we could see certain things starting to really jump off the page. And the so-called ID8, which is the kinds of deletions that are occurring involving the number of repeat units and the size of them, they were substantially increased, much higher than you would expect in the normal background. And similarly, microhomology deletion length. So when we looked at this and analyzed this against the radiation exposure, increasing radiation leads to more of these small deletions. When we did that in toto, looking at the total number of deletions, and then also using the SNV ratio, the deletion to SNV ratio, as a mark of how long these tumors or how old they were, we could see the effect was even stronger. We know that the majority, over 70% of the SBS background signature of SNVs are the clock signatures, which are really an indication of just steady deterioration and breakdown, or rather substitution of a base at a time. Clock signatures can be very useful for timing a disease, together with new mutations or a second driver or the advent of SCNAs. But since we have so few SCNAs, we couldn't really time in that sense. This was interesting because high-dose therapeutic radiation, there had been a paper by Sam Bajati and company a couple of years ago on some 12 tumors. They saw an enrichment of deletions more than insertions in microhomology of those junctions, just as we had seen here. We have 400 and some tumors to look at. They had 12, but they at least were able to see something coming off. And then just a week ago in Nature Genetics, Flores, Bartel, and Roel Verheij published this very interesting paper of looking at radiotherapy in particularly brain tumors and some other post-treatment metastatic tumors. And they saw a substantial enrichment in the small deletions and large SVs in genomes exposed to high-dose radiation therapy. And cosmic indel signature 8 was the primary thing that they saw. And it was comparable effects of radiation across the genome. The dose and amount and intensity influenced that extent. And so here we had, remember, relatively lower doses on the average, 0.5 gray over a matter of weeks, as opposed to people who are having radiation therapy where they may get 5 or 10 gray to a very discrete part of the body, and it's all over 2 or 3 weeks. But it's very interesting because their study particularly recapitulates what the animal literature, which there's quite extensive information on. So we then went a little bit further, and we published a couple of years ago the relationship between radiation dose and gene mutations in post-Chernobyl thyroid cancer with Yuri Nikiforov. And we had looked at 65 individuals by RNA-seq and saw that a lot of them, we saw an enrichment of fusion proteins per se, in a few point mutations, but this was a biased group that was picked of high dose and particularly young, but this was a very informative sort of preliminary analysis. Well, when we looked at our drivers, we could identify a candidate driver for 98.4% of the tumors. Remember before I said we were about 80% with TCGA. And interestingly enough, 93.5% of these are in the MAP kinase pathway, which we knew was important from TCGA, but we didn't know that it was this important, particularly in younger individuals in the Chernobyl setting. So the 253 mutation drivers, we could see BRAF and NRAS, KRAS, and the like, and a little bit of DICER1. But for instance, we really didn't see many TERT mutations. In fact, we only saw one total, which was quite different than what you see in the adults. We saw a lot of fusion drivers. This is all the group put together. And you'll see when we start separating and looking at the higher doses, you have a higher and higher fraction of the fusions. But these are, by and large, other than PPARG, and genes that are critical in the MAP kinase, the mitogen activated protein kinase pathway. And so there's a kind of parsimony that we know that thyroid cancer is, particularly papillary thyroid cancer, is really taking advantage of. And so we saw those particular genes in the PTC, and then a few of the ones that were not there, like structural variants in IGF-2 BP3 and IGF-2 are each, as you can see, very important in their interaction, particularly with that MAP kinase pathway. So something about the development of the thyroid in its very isolated and in its isolated sanctuary becomes particularly interesting when we think of what the drivers are and how they arise. Okay, so the TERT mutation, which was 20% of the TCGA cases, we saw one case in our younger Chernobyl study. Okay. We looked at the thyroglobulin, which is a gene frequently mutated in thyroid tissue, but it's not a driver. And this gets at a really important question that we need to start to look at informatically. Insertions and deletions target lineage-defining genes in human cancer. So going and looking at particular genes, you could say, all right, this is important for thyroid, papillary epithelial development, which we know TG is. And you could see many, many more mutations than in BRAF, but almost all of them were non-coding. In other words, they were intronic, as you can see there in the right column there. I mean, it just tells us of where some of these events are taking place. Now, whether they're early or whether they're at once the cancer starts is still a debatable question. So when we looked at the PTC driver, the shift to fusions with radiation exposure, I told you that we saw that increase in fusion drivers. And if we include indels in the structural variants, which I'll talk a little bit more about in a minute, we can clearly see a very strong effect. And we actually look at the excess odds ratio. So in other words, as you go add 100 milligray to each group, so to speak, if you distribute them over the amount of exposure, the higher the exposure, the greater the likelihood of developing fusion events per se. And we didn't see any clear single radiation biomarker that was occurring over and over per se. So for structural variants, which we know are one of the more challenging things to pull out of genomes. We use multiple programs to do this, and had to do some extra sequencing to be sure that we understood that. We know about deletions and reciprocal inversion, foldback inversions, unbalanced translocations, chromoplexing, chromorthipsis, people's favorite forms of complex. And so when we looked at this, we could see that radiation exposure increased the number of structural variants that we're seeing, particularly of the simple ballast version, which was by far the most common. In other words, where you saw one event moving to another and you didn't have 2 or 3 with directional changes. So we know that DNA double-strand break repair mechanisms are very important, and there's been a lot of literature on radiation of cell lines in animals that clearly suggest that there are particular mechanisms, such as the non-homologous end joining mechanism, but no one had ever seen this really in human tumors and studied this. So we were able to look at actually each and every one of the structural variants, as well as the fusions we identified, and look in the DNA sequence and ask the question, what does the immediate sequence informatically tell us or teach us? Does it illustrate a particular type of event taking place? And in fact, the answer is an emphatic yes. We knew that there are very different distinct mechanisms for high fidelity and large deletions, with SSA, for different types of repair mechanisms. So when we looked at ours, we could see that the five base pair deletions and microhomology were associated with dose, particularly strongly when it was, the shorter ones, 1 to 2 to 3 or whatever. And you could see this effect with radiation certainly increasing. So it really, in our minds, we could use the sequence to really gain an important insight into exactly how we think this is causing. So remember, radiation, ionizing radiation, is a class 1 IARC carcinogen. Exactly how it works, we now have a much better idea because we now have very strong human data in these 360 cases of radiation exposure. Remember, there were 80 that had no radiation, and we can see this relationship really holding up very well. So non-homologous end joining is the primary double-strand break repair mechanism that's affected by radiation. So we also know that radiation effects vary by age and exposure. Stronger radiation effects at younger age. We could do that when we started breaking down our data set and looking at less than 5, 5 to 9, and particularly greater than 10, see those two extremes looking quite different. We then looked at 22q, which has this loss of 22q that had been described as a driver in TCGA. And in our setting, it was about 18%, but it was strongly associated with RAS mutations, but not BRAF. And we didn't see it as a singular driver. We think of it as a secondary kind of driving event in a subset, particularly of the RAS mutation. Okay. We then looked at germline, in which we saw few had a very rare damaging variants in cancer predisposition genes, but we didn't see a plethora of any one particular mutation. It's no different than what we would see if we'd sequenced 500 children with pediatric cancer. But what was interesting was we looked at a polygenic risk score based on 12 previously reported SNPs that the DECODE group had identified. So an interesting inverse relationship that the PRS effect was higher if it was a lower dose. So in other words, radiation would supersede whatever and drive the cancer, as opposed to the higher PRS may be for individuals who are not developing radiation-associated cancer. And so the higher genetic load may be contributing to their risk for the sporadic background thyroid cancer. And this was an intriguing answer, a finding that we have to go further on. Now, we had methylation data, or the patterns in the Chernobyl tissue cancer. The driver, really the types of methylation patterns were not in any way related to exposure. When we did these same graphs by putting out, by 0 to 9 gray, no effect that we could see. What we really saw when we were looking at the tumors is that it's the driver mutation, whether it's a BRAF or a RAS, that really separates. So in other words, the characteristic driver, the parsimony of that driver is driving these events. So you can't say that, oh, we see a very clear kind of radiation exposure in these tumors per se. There had been one gene CLIP2 that a group in Germany had published on with using samples from the Chernobyl tissue bank, about 50 samples, which overlapped with ours. And when we almost, did 8 times that, we saw 0 effect. Others had not seen anything before, but this has been maintained as a possible CLIP2 as a biomarker for radiation. We see no evidence for this in the tumors. And then we have about 200 non-tumors, in other words, thyroids from individual where they were taken out, and we looked at the normal contralateral thyroid, and we don't see CLIP2 in any way as a biomarker for radiation injury. So to sort of sum up this part, we certainly know about April 26th and the contamination of the environment. Double-strand DNA breaks, clearly how we see the cancers were actually caused. The thyroid cancer, then leading to an understanding that we have from the genomic landscape of looking at these double-strand events as a function of radiation. So our key findings are that it's a very parsimonious tumor with 93% of the mutations in the MAP kinase alone. The importance of the double strand, talked about that. Epigenetic and gene expression profiles are determined by the cancer driver mutation and not necessarily by the radiation exposure. So we have almost the same number of FFPE samples from the Chernobyl accident with a greater number of high dose that we're now looking at. Analysis of multifocal. We have some circumstances where individuals have more than one cancer, and the first handful of them are really suggesting that they usually are asynchronous and not the same driver mutation. And the analysis of metastatic tumors we're just about complete with, and we can see the same driver in all the metastatic tumors. And the question is, what changes with the metastatic lesions? We're working through that. We're looking at the field effect, so to speak. We don't see radiation signatures or drivers in the companion non-thyroid tumor, but we have more work to do on it. We had that in our paper, and one of the reviewers asked us to take it out and suggested more rigorous ways of approaching that, which we are now looking at. With these greater numbers, we can look at this question of age and PTC and pathologic characteristics, and now we're expanding to other radiation-exposed tumor types and populations. So Lindsay Morton and Danielle Cariotti and Eric Dawson, and of course our colleagues, academician Tronco and Professor Bogdanova, were really crucial in being able to do this. And of course, our longstanding collaboration with Gady Getz and Chip Stewart at the Broad. So I'm going to switch gears now for the next 15 minutes to talk about story number 2 in Chernobyl. This is transgenerational studies following the Chernobyl accident. So first question is, could there be a transgenerational effect? This has been a profoundly important political issue. If you look at what happened in Fukushima, the nuclear reactor that was affected by the tsunami 10 years ago, one of the 3 major reasons why people are not moving back into the area is that particularly families of with potential childbearing age are worried that they could have an effect on the subsequent generations. Early literature suggested microsatellites in small numbers of 10 or 15 PCR-based assays in children from Chernobyl. But in Nagasaki, there were 3 family trios that had whole genome sequencing, but no observed unusual effects per se. And we wanted to know, was there a transgenerational effect? In other words, something that would happen to the egg or the sperm, and then subsequently in the second in the children, that would be seen that would be different from the parents. Okay, so it's really focused on de novo mutations, which I'll talk quite a bit about. So we created a family structure study. We've recruited even more, but we're publishing on the first 105 families with 130 children born between 1987 and 2002. So they had to have left, and the first child is born at least 46 weeks after leaving. So in other words, no conception while they had active radiation exposure. We did whole genome sequencing with short read technology at 80x coverage, not 20 to 30x, but 80x. We wanted to be sure we did not miss anything. And we used multi-step calling algorithms and manual inspection of all the putative events. So our study design was published in the American Journal of Epidemiology with academician Bazika from the Ukraine. Is the first author of that. We know that there are multiple transmissions of de novo mutations. And the Icelanders had published elegant papers, as had groups here in the United States, that show there's an average of 40 to 70 de novo mutations per generation. And the single largest, most important factor is paternal age. As father's age increases, the number of DNMs increase by maybe a DNM per year or so. So DNMs are essential types of mutations. They're due to random mutations in the gametes, post-zygotic spermatocytes, and a small number of variants are generated. They're transmitted, are known as de novo mutations, the critical block of evolution because there's no purifying selection going on here. Okay, this is what's happening randomly in the genomes of spermatocytes. They account for a fraction of neurodevelopmental disorders, pardon me, such as autism and some of the catastrophic pediatric diseases. And they're ascertained by analysis in which you have to have both parents and the children, and you see the variant only in the child, but not in the parents. This is not like clonal hematopoiesis or anything. It's a long-standing controversy on a possible transgenerational effect due to radiation. Previous high doses of animal studies had suggested that, showing NHEJ, DNA repair, with structural and chromosomal events, but no single base mutational signature. Humans have lower protracted doses, smaller underpowered studies, such as with the microsatellites in the Nagasaki that I mentioned before. So we also spent a lot of time looking at the cleanup workers. So here is the American-Ukrainian trio study of parental radiation of cleanup workers and a few evacuees. So extensive radiation estimates were done with the dosimeters to understand exactly what each person was exposed and for how long, so that we could then look at the principles of the DNMs and understand them against the radiation. So here's the principle of how we find the DNMs. So we had 80x coverage. Maternal variants and paternal variants were looked at, and we looked for variants that were inherited from the mother or the father, 99% of those. But when we saw what traditionally would be called Mendelian inconsistency errors, we see a new variant not in either parent. And then there was an ability to go through these and determine whether they were sequencing errors, asking questions of filter depth, quality, probability, and we would get down to less than 200 that we actually, for each child, we hand annotated in IGF files, going in and looking, saying, could we see this? And then going to the parents, saying, we don't see this in the parents. So this is a bona fide de novo mutation. We had adequate power based on a linear dose model with a two-sided test conducted at 0.05 to see if they were 0.6 DNMs per 100 milligray of father's dose or 5.5 for the mothers. We know that much stronger effect from the father, but a small but real one from mothers. So here is the key thing. When we then made our tables and looked at the DNMs in each families, and then we looked at the different variables, we could see that the maternal age was just above significance, but the paternal age tells us very strong. So 3.65 times 10 to the minus 15. There's no question that the older the father, the greater the number of DNMs. When we looked at the radiation dose, the cumulative radiation dose, either the maternal or the paternal, we saw nothing significant by p-value or by estimate. When we looked at smoking, we could see that former smokers had nothing. Current smokers, there was a borderline question with the mothers, but the literature has looked at this in other studies, like in Iceland, and not seen anything. So we don't consider that to be the final answer there. So then we looked at the parent of origin estimates. So about 42% of these, we could see adjacent SNPs around these that we could track and say, aha, this mutation is, we could see this is happening on the background of the allele that the mother had transmitted or the father had transmitted. And here we can see separate models were fit to estimate the paternal and maternal effects. And the paternal effects, you could clearly see strongly there, but the maternal now emerged to be quite a bit stronger when we could actually get to the maternal origin of those DNMs. So then we looked at the question many different ways because we were a little surprised by our findings. So we look at the cumulative dose. We look at the cumulative dose truncated at 1 gray or 1,000 milligray, no difference between mothers and fathers, cumulative log radiation dose, and we had 3 or 4 other models that we proposed, but we could not find a way to make our data significant. We know that repetitive DNA in the genome is probably the primary target for DNMs, and this comes from Steve Salzberg's marvelous review that's 9 years old, but has basically been held up and looking at the distribution of these kinds of events and the frequency. And there's some 425,000 mini micro satellites. And so did we see something there? Or did we see these events taking place in lines and LTR retrotransposons? And the answer is interesting, no, no, and no. So if we look at the different types of events, starting at the bottom shows the total DNMs, which I've already shown you that. If you look at SNVs, there's no difference with radiation dose. If we look at microsatellites, there was nothing significant that we saw. We identified 730 microsatellites. Indels, 2,000, complex and clusters. And there's a subtle difference between complexes and clusters we can talk about a little bit later. But what was interesting, we just didn't see effects for any of the subgroups. Now, there have been these anecdotal reports, I didn't put them in the talk, but of people saying, we have a patient who was from the Ukraine and exposed, and parents were exposed, and they have this strange complex in which you see 3 SNPs in a deletion maybe 200 bases away. So that with an event that we assume occurs all in one generation, so to speak, that it's not a cumulative somatic type of event. So when we compared across all the other studies, when we looked at our numbers for instance, the SNVs, we saw no difference than what had been seen for the total number or the distribution, the types of events that had taken place in the different, the different possibilities of C to A, C to G, C to T, T to C, and the like. And so to us, this was very reassuring that there was no selective process going. So different genome builds, pipelines, and validation means that we can only compare this to a particular point. And it's really important to recognize you have to call everything at the same time. Genome builds and genome calling have small errors, but since what we're talking about is with the DNMs just going back, literally 9,000 SNVs in 130 individuals, 730 microsatellites. If one is going to compare these, you have to have called the BAM files identically. We have some people already who are saying they see findings in our study, but it's because I think they're sloppy. They're not doing what you need to do rigorously, and that is to call everything in parallel. So our study considerations are looking at a peripheral blood of adult children. We are not looking at those horrific events that may have occurred because people were pregnant at the time or had very high exposures with the acute sickness who did not survive. But we did not see an increase in DNMs in children born in 1987. There's a survivor bias for sampled children recruited as adults. We recognize that. And we didn't have a lot of high acute doses, but the likelihood that those people would go on and father or be parents is relatively small. We now have a few twins, but no twins in this particular study per se. So we see this as a very important circumstance of where genetics and public health intersect. We have adequate power to identify substantive increases in DNMs in adult children born to liquidators, and we don't see that. And this is important because if you go back to Fukushima, which, remember, this has been a big issue, and this paper has gotten a lot of press in Japan, really, the Fukushima levels were much lower than what we're talking about with the liquidators and the evacuees in Chernobyl. This should be very reassuring to those families that it's all right to go back. Doses were both extended lower than in animal studies. That really gets us this interesting question of why there are lower gonadal DNM events. That altered balance may be more favorable for a certain amount of repair to take place when the radiation is exposed over time, as opposed to one very, very intense dose of radiation. So we saw no evidence of a radiation-induced single-base mutation or epigenetic signature in this. We did the epigenetics. I didn't show that here. Adequate power to identify substantive increase in DNMs. And we have further trios of high dose that we're going after. And we had a long-term conversation with the survivors of the Hiroshima and Nagasaki blasts. And we also are right now investigating clonal hematopoiesis and genetic mosaicism in the parents with respect to radiation dose, given a number of papers looking at those kinds of events being increased and in the course of exposure to radiation. So with that, want to really call out the marvelous work of Meredith Yeager and Mitch McKeela and Amy Barrington in our program, and then of course Dimitri Bazika in the UK, who directs the NRCRM, which is the National Research Center for Radiation Medicine of the National Academy of Medicine in the Ukraine. And lastly, let me just show to the trainees here, I'm the director of DCG, where we have 71 PIs and about 130 trainees at any one time. Spectacular opportunities for postdoctoral training and beyond, and lots of different things that we do. And hopefully there are some things in those circles that may be interesting. And if you're interested, we are very interested in training people, and there are remarkable opportunities that we have. So with that, why don't I go ahead and stop and let's see where we are. Let me pull up, I got to pull up the questions.
We've got some in the chat too.
In the chat, okay. Well, I don't see open questions, Natalie. I just see in the chat. Okay. Okay. All right. Thank you. Okay. Methods for integrative omics data. Yes, our data are now in dbGaP. It will probably be in July that they'll be more easily accessible. You can go to the Genomic Data Commons, and each of these two studies have study pages. You could get the BAM files today, going through dbGaP. But if you want any of the more refined files, that will be in July, simply because the way the Genomic Data Commons does its own it sort of uniformly sets all of the data sets. And so there'll be a July release. To be honest, all of the outcomes are in the supplementaries of our two papers in Science. So you can go and you can see every DNM is listed, all right, every single one. And for the second study, and for the first, every somatic mutation is identified as well. So you can see that by individual. For the DNA. The RNA, you can see a certain amount of the expression, but if you wanted to redo the RNA expression, I mean, you really need, you need to go get the BAM files and call them the way you'd like them, okay? All right. Nadia, I see you have a question about genetic mosaicism and how would you investigate it? Yeah. So the first study, we didn't see much of anything. It's really in the second one where we have individuals who are presumably healthy. As I should have said, virtually none of the parents and children had cancer in the second set. And so these are individuals who were there, left, and they go on with their lives. And the question of mosaicism in clonal hematopoiesis is very pressing. As you know, it's age-related. And the question is, these individuals are now 35 years out, and they were 20 years at the time, so they're 55. They're in the sort of the peak time for the takeoff for genetic mosaicism and clonal hematopoiesis. And the question is, to us, looking at the parents, do we see an increase in clonal hematopoiesis and or genetic mosaicism? So we have the whole genome sequencing, which we're looking at very closely for that, with ADX. And then we have chip data. a SNP chip data. And we're probably going to do some targeted sequencing at like 500 to 1000x to see if we can see, the early signs of faster liftoff for TET2 or DNMT3A, some of the genes that are classically seen in as early, as sort of the early pioneers of clonal hematopoiesis, as sadly our genomes fall apart. That's a whole other talk that I'm happy to give, but not today.
Thank you. And I guess another question I had that I was wondering about as you were speaking for the second part of the talk, how difficult was it to recruit parents and their children for this study and to convince basically 3 members of a family to participate? And how did you go about doing that?
Well, so the NRCRM in the Ukraine, led by Dmitry Bazika, is a special institute. I've been there physically several times, devoted to the long-term care of the liquidators, the Ukrainians who are part of the cleanup. And so they have a very good relation. We wrote a protocol 8.5, 9 years ago, and then Dmitry and his staff went and they then came up with the approach of how they would introduce this to their to individuals. And they started by, we wanted to be sure that we had individuals who had high dose, the mother or father, both or neither. And then they contacted, and we had remarkably high acceptance rate. But again, we have to recognize there's a certain bias, because these are people who have survived. They have not had horrific health conditions in the first 5 or 10 years. And so they were interested, because this is a very important question. Are their children going to have problems? So once that was presented, people were very interested, because it's, I'm 55, I'm such and such, but boy, I want to know, is my 20-year-old son going to be all right, or my 19-year-old daughter? That's what I'm really worried about, because they're going to have families. Does this mean they're all going to be terribly mutated, like, in a science fiction movie or something?
And we do have a comment and a thanks in the Q&A.
Mm-hmm. Yeah, I see one from an individual whose family member was actually present in Chernobyl and ended up with a tumor the size of a football removed from the abdomen 15 to 20 years later. Yes, we know that solid tumors certainly can be seen, but they're often of lots of rare variant types. So it's hard to say. We know in terms of the radiation-related issues, leukemia and aplastic anemia occur in the first 5 to 10 years, and then they gradually decrease. Unfortunately, many of those are fatal. And the question is, the sarcomas or the sort of quasi-benign large growth. And then the question is, at what point does breast cancer or prostate cancer or lung cancer, colon cancer increase or not? And those are pressing questions that in this particular NRCRM is being looked at right now with, thousands of people who are being followed to see if indeed we do see a shift in that. Okay.
There's another one in the chat from Min. There is no correlation between radiation dose and DNMs. Is this maybe because radiation is causing mutations that are not detected?
Well, yeah, that could be the case, but they would be, I tucked into one of the slides was, we did SNP chips on everybody as well. So if there was a large structural event, we should be able to pick that up. And we did not see that. Do we need to do PacBio sequencing? We've thought about that, and we probably will in an exploratory way. But with short read ADX, the likelihood that we're missing very large events is relatively small. I think we know enough about using panels of normals to be able to do this. So we actually have taken our genome sequence, and for the DNMs, we played hide this sequence. So let's just go with randomly 30 of the ADX coverage, and what does that get us? And the small but real difference is there, okay?
We have a couple now in the chat.
Okay. Is there a function of the nature of radiation is similar increases in incident hasn't been observed in inhabited areas with high background radiation? Well, the thorium deficit areas in India with people living there for generations, that's a really interesting question. I have to say, some of us, we know about that. We're still somewhat skeptical about the ascertainment collection of information there, okay? That's an area that there's been a lot of discussion and doing a more rigorous approach towards. In Brazil, there are mining areas where they've had areas of either accidents and or high ambient radiation exposure, but usually those aren't quite at the level that we're talking about here, okay? But it's a really important question. Okay. So any single cell data on father's sperm for de novo? Not yet. Again, Nadia was asking me about compliance. Well, the original protocol, I had asked whether we could get blood, skin, and sperm. Well, we could get blood, we couldn't get skin, and sperm was just completely not possible. That was, no, I mean, there are cultural and all sorts of reasons. I think now we may be able to ask in some circumstances, but the issue is most of those individuals are much older now. You know, I mean, it would be interesting, but these are people whose children are now 25 years old, and I'm not sure how well we can extrapolate that, okay?
Are there any other questions?
Well, it looks like the chat box is complete, and hopefully.
Oh, I don't know, Jasneet, do you mean to have your hand up? No.
Okay. Well, great.
Really fabulous talk.
Well, thank you for listening. And, you know, the story goes on. There'll be more chapters to this, believe me. All right.
Yeah, thank you so much for your great talk. And yeah, we're looking forward to use some of this data for the analysis.
Absolutely. Please don't go do it. That's the whole point. Okay.
Yeah, so
And
wait for
when and if you find something interesting, please let me know. Love, you know, we want to know how this data lives on and what the conclusions are or not. Okay.
Yeah. So in addition to all these omics data that you mentioned early on in your slides, so like for the patients and also the controls, say demographic and other clinical information, they are all available on DVGAP?
Yes, there's a certain amount. So you'll know the age of the person at exposure, the age at diagnosis, the sex, which oblast, in other words, which like the equivalent of a state that they lived in. And then we have further information that is not yet posted about survivorship and treatment and things like that. But that will come a little bit later, okay? I mean, we're working on that right now, and our Ukrainian colleagues don't want us to release that until we finish that work, okay?
Okay. Thank you.
Pleasure, all right?
Yeah, thank you so much for your time, especially right after your surgery last week.
All right.
And we
Pleasure.
have a comment in the Q&A. So thank you, very nice presentation with a very nice voice also.
Thank you. All right.
Okay, yeah, thank you, Dr.
All right.
Chanock, and thank you all for joining us today.
Stay well and work hard and integrate away. Okay. All right.
Thanks.
Bye-bye.
Bye.
Bye-bye.